Hello, my name is Dr. Roderick L. Rowe, and today I will be presenting on the antigen presenting cell. A molecule that triggers a specific immune response is called an antigen. So now we're finally at the third line of defense. This is also called the acquired immunity, and it is going to involve the B cell and the T cell. But before we can get those activated, we have to devour the thing that's foreign, and that foreign substance is the antigen. The antigen can either come from outside or inside, so it can be either exogenous or endogenous. Exogenous antigens come from outside of the body. For example, in the picture, we're looking at a bacteria and a virus. The virus is a little small circle that has the landing gear, and I'm pointing to the landing gear. So all of that is foreign to our body, and it came from outside. It is the exogenous antigen. Exogenous antigens include toxins and other components of microbial cell wall, cell membranes, flagella, and even the sex pili. This slide is showing you a gram-negative bacteria. The gram-negative bacteria has a portion of the wall that would be an exogenous toxin. I'm pointing to the lipid A, very dangerous. The other type of antigen is the endogenous antigen. It comes from inside of the cell. Right now, we're pointing to one of those landing gear, maybe the wheel of an airplane. So I always explain the virus as a airplane with tires, and the tires are part of the landing gear that have to be deployed and touch the road as it is landing. The road is your cell membrane. So in order for the virus to penetrate, to get inside, it has to first bind to your cell. It has to land. So this image is showing you that the virus, this intracellular virus, has hijacked your enzymes that are involved in transcription translation and has produced its own landing gear, its tires. And then now your cell uh, will produce and put the tires for the virus on the surface of the membrane. That is an endogenous antigen. It came from inside of your cell. So these endogenous antigens are produced by microbes that reproduce inside of your body's cells. A viral particle expressed on the membrane is the scenario I just went over. So we're still on the third line of defense. A macrophage works with the acquired immunity. In the picture, I'm showing you in a macrophage, and this macrophage uh, is going to devour one of those um, exogenous or endogenous antigens. To illustrate this, we're going to talk about push and pee. Push and pee is just a cool way to explain the three P's. The three P's of how the macrophage is involved is preparation, processing, and presentation. So let's start with push and pee. Preparation. The macrophage must make receptors, pushing P. So preparation of adaptive immunity involves making the receptor. So in the picture you see the antigen presenting cell has made a receptor, and I'm pointing to it in the picture, and that receptor is MHC. There's different types of there are different types of macrophages. There's different types of antigen presenting cells. There's different types of cells that devour foreign substances. So antigen presenting cell can be separated into a professional antigen presenting cell and a non-professional antigen presenting cell. The professional one includes B cells, macrophage, and dendrites. So I might just refer to them as APC. There's also some non-professional antigen presenting cell, microglial cells in the brain, and these Kupffer cells in the liver. We are going to specifically talk about the professional antigen presenting cells, which will include the ones I just discussed, macrophage, B cells, and dendritic cells that are located in the skin. And these antigen presenting cells will make a membrane receptor. These membrane receptors are called the major histocompatibility complex, or MHC. In the picture, I'm pointing to the MHC. I think of the MHC as a glove that you put on. And once you have the glove on, then now you can capture something. So in the picture to the left, you have a glove that we're pointing to, MHC1. In the picture to the right, you see another glove, MHC2. The glove, the MHC molecule is a glycoprotein human cell marker. That marker has, or glove, has two purposes. It helps to identify self, and it holds the epitope. So I think of it as a glove because it's holding that epitope. It's holding the thing that is foreign. Remember, the exogenous or endogenous antigen can be broken down into an epitope. So it can be held in that glove. I mentioned the MHC molecule being involved in recognition. So where did that come from? How do we know that it's involved in recognition? It all started with the skin grafts. So I want to take a brief moment to talk about a skin graft. So animal skin grafting, or xenografts, involves using skin from another animal species like pig to cover wounds or burns temporarily until the animal's own skin can heal. So in the picture I'm showing you where some catastrophic event occurred to these animals where their skin was damaged by a burn. And the doctors have surgically placed another animal type, another species over that area to help it allow to heal. Now that area is going to be prepped so that blood vessels will now be able to supply the nutrients to that new skin. Here is before, during this process, and finally the after. Scientists discovered if the animals were not closely related in skin grafting, the recipient rapidly rejected the skin. This rejection is due to the MHC complex. not being identified by self. So originally we talked about the purpose of MAC was to be to hold the epitope and for identification. So this was discovered by these scientists when they were doing skin grafting, that if the species was not really close, you would have more of an issue with self-rejection. So what makes those receptors that helps the body identify self? So a cluster of genes located on chromosome 6 will make these receptors. And that glycoprotein receptor is separated into three classes, MAC1 and 2, which have the broadest binding capacity, and MAC3. MAC1 appears on all nucleated cells and not on red blood cells. Red blood cells do not have a nucleus. Therefore, it doesn't have that chromosome, it doesn't have that gene, and it cannot produce this glycoprotein. Endogenous antigens are processed by antigen presenting cells, and the epitope is presented on the MHC class 1 receptor. Look at the picture. I know it's sideways, but you still should be able to understand it. This is when an endogenous antigen is detected within the cell. So number one, a polypeptide is processed. They may see a protein in the membrane is located on the endoplasmic reticulum, and we know that commonly is called the ER. So the MHC1 is on the ER, and it is going to bind to that epitope when the epitope comes into the cell. If you look at the image label 3, the ER then sends that package to the Golgi body or Golgi apparatus, where it is still attached to the MAC1 that was originally on the ER. Go to image 4. Vesicles fuse with the cytoplasmic membrane. So the MAC1 now puts it on the cytoplasmic membrane. So now you should understand uh, the endogenous antigen picture below now. So red blood cells do not have a nucleus, like I mentioned, so they cannot produce those MAC1 receptors. Well, they can't produce MAC receptors, period. I'm not saying that you cannot have rejection of red blood cells. You just can't have the rejection of red blood cells via that glycoprotein called MAC1. If red blood cells are infected by an intracellular pathogen, it will not be recognized by T lymphocytes for the adaptive immunity response. So when those infections occur, like if we someone has Plasmodium vivax, which is the causative agent of malaria, when that uh, protozoa goes into the cell, it is not going to be a particle of it is not going to be bound to the MAC1 receptor. And that's what this slide is showing you. So those individuals that have these intracellular infections of the red blood cells, um, it's hard for the immune system to identify it. So what happens is those individuals have these undulating type symptoms. where it goes up and down, meaning they're sick and then they're not sick. They're sick and they're not sick. Well, when they're sick is when that those cells are going to burst out of the red blood cells. But when they are in the red blood cells, they are not observed by the immune system. Here is some slides that show red blood cells and how they have a intracellular infection. Schizozoints is one version that you'll read about when we talk about malaria, where that protozoa is in a specific stage of development and it is taking place inside of the red blood cells that are in this picture. Everything that you see purple within a red circle, that is the schizozoint stage of malaria. This slide here illustrates it in much more detail if you want to know about how the red blood cell gets infected. Start with on the human liver stage side, A, go through 1, 2, 3, 4, and 5. That shows you where the protozoa enters the red blood cell. So that was MHC1. Now, MHC class 2 is found on some types of professional antigen presenting cells like the macrophage, dendrite, and B cell. Here is the dendritic cell. We're going to talk about it extensively. So you need to know that it is located in the skin. It's also called the Langerhans cells. So dendritic cells usually process the exogenous antigens and presents epitopes on MHC class 2. So how does that work? So let's look at the picture. I know it's sideways, but you can still see the macrophage to the far left devouring this bacteria. It's going to devour the bacteria and it's going to process the bacteria. Notice how inside of that macrophage, you have a lysosome, and the lysosome has MHC2 receptor attached. You go down to the next image, image 2, you know that the phagosome is going to bring the antigen in, and now it fuses with the lysosome. When it fuses with the lysosome, now it is a phagolysosome. This phagolysosome has the MHC2, then a particle, an epitope, is going to be attached to the MHC2 glycoprotein. Now, through exocytosis, uh, this vesicle fuses with the membrane, now exposing the MHC2 glycoprotein with the epitope on the surface of the antigen presenting cell. So I showed you so far two methods, endo and exo. I'm sorry, the endogenous antigen versus the exogenous antigen being processed and exposed on the surface of these cells. We're going to skip class 3, but it is a protein that is involved with the complement system. Pushing P is specifically talking about preparation of those receptors. So I'm finished covering the first P. We talked about MHC1, we talked about MHC2, and we just concluded with MHC3. Pushing P. What will the macrophage do? So we're going to show you how it processes the foreign entity. The first part was just really basically about those receptors, MHC receptors. So circulating antigen presenting cells encounter a foreign antigen or cell and devours it through phagocytosis. The picture is showing you how this occurs. And it's 5 steps, starting with the chemical attraction, ending with exocytosis. The processing of the antigen occurs within the nearest lymphoid tissue. So after the phagocyte devours the foreign substance, it's going to move through the bloodstream or lymphatic system un- until it gets to the nearest lymph node. It brought in that foreign substance, antigen, and it brought it in through a phagosome. The phagosome fuses with the lysosome. The phagolysosome is then going to process the antigen and expose a part of the antigen called an epitope. And in the bottom picture, I'm showing you how the epitope is just one piece of the antigen. Pushing P. That was processing. Processing is to devour and fragment the antigen into the epitopes. We now look at presentation. Pushing P. APCs have both MAC1 and MAC2. The professional antigen presenting cell may have 100,000 MAC2 molecules on the cell's membrane, which binds thousands of different epitopes. After processing, an epitope will be inserted on a clift on the major histocompatibility complex 2 of the antigen presenting cell through exocytosis. So I'm showing you the macrophage processing, and it will expose that epitope on the glove. And the glove has a cleft, and I'm pointing to the cleft in the picture to the right. Here's another picture to show you how it was exposed on the cleft. It was first processed, fragmented, and now elimination through exocytosis. The presentation of the epitope uh, to the T lymphocyte triggers the immune response. So now that the antigen presenting cell has the baseball in the glove, it is floating around in the lymphatic system or the bloodstream, holding this ball in the glove. When it binds with a T lymphocyte, it will create what is called a immunological synapses, the joining of two hands, because the T cell has a receptor as well. So when they bind together, as in this picture I'm pointing to with the red arrow, and an immunological response occurs, now we are finally at the acquired immunity. We're now at the third line of defense because now the T cell is involved. If the epitope does not bind, it will not trigger an immune response. So again, the APC has the catcher's mitt with the ball and it's floating around. And if it does not bind to a T lymphocyte, then you will not get an immune response. So epitopes will bind in some people, but won't bind in others, which is the foundation for allergies. So if you don't have allergies, you have APC cells floating around with balls in their catcher's mitt. They have processed an epitope, but your T cells do not recognize it as being foreign. It doesn't recognize it as being uh, detrimental. So it will not bind to create the response. Some people, it will. Push and P. So we talked about preparation, processing, and presentation. Presentation may or may not trigger the response. So MHC1 and MHC2 both have broad binding capability. The binding diversity is dependent on an individual's gene. This concludes the presentation. Make sure you understand all of this that's in the slide. Preparation, processing, and presentation.